So hello and greetings to everyone. This is Vaibhav Shukla from Vaibhav's Biotech Odyssey channel, and I hope you all are doing great. And today in this video guys, we are going to have a discussion on the topic that is CAR T cell therapy. So the abbreviation CAR means chimeric antigen receptor T cell therapy. Okay, so the word chimeric itself means fusion or hybrid. Now, this CAR is designed to bind a specific antigen, right? That is a molecule on surface of cancer cells that the T cell will be going to target it. So let's get a look onto the steps, that what are the steps involved in this process of CAR T cell therapy. And to must remind you that this therapy is used for treating the diseases like cancers. Okay, so in the first step you have to take the blood sample from the patient, and you have to you can say you have to isolate the white blood cells, or you have to specially isolate the T cells from the patient's blood. Okay, and then you have to return the blood back into the patient. Okay, now after this you have to modify the T cells by altering the genome of the T cells. Okay, this therapy basically increases the, you can say, efficiency or the effectiveness of the T cells to destroy the cancer cells more effectively as compared to the normal one. So in the second step, as you can see in the diagram, uh, the CAR gene has been added to the genome of the T cell with the help of a viral vector, or you can say the virus. So as we are using, you can use any kind of viral vector like lentivirus or retrovirus for effective transfer of your gene, CAR gene, into the genome of the T cell. So as you know, the basic concept of recombinant DNA technology, that to transfer the gene of interest into the host cell, you must need a vector. Okay, so here we are using the viral vector to transfer that CAR gene into the genome of the T cell and so on. Okay, so after the insertion of the gene, CAR gene, into the T cell, now your T cell has been modified, and due to this expression of the CAR gene, the CAR receptors will be expressed on the surface of the T cell, or you can say it will be formed on the surfaces of the T cell. And then these receptors are specific to the cancer cells. That means they are going to only bind to the targeted cancer cells only. Okay, now after the modification you have done in the lab and everything, then now you are going to make the multiple copies of that T cell, or the million copies of that T cell. And then after the multiplication process, you are going to reinsert those modified T cells into the patient's bloodstream. Okay, now after this process, what occurs in the body, now these cells will go to the, you know, these cells will specifically find and bind to the cancer cells and will ultimately destroy them. Okay, so this is the whole process of the chimeric antigen receptor T cell therapy process. I hope you understand this. And in the case of the genome editing of that T cell, you can also use the genome editing tool like CRISPR-Cas9, which is a very precise gene modification tool. Okay, as it prevents the off targeting. Okay, so I hope you have understood the whole concept of the CAR T cell therapy process, and the cost of this therapy process in India is approx 40 to 50 lakhs, right? So if you like the concept, make sure to subscribe and share the video, and subscribe the channel as well. And if this video reaches 100 likes, then I will make a video on the CRISPR-Cas9, where I'll be explaining the full concept of the CRISPR-Cas9. So thanks for watching guys.
お前はもう死んでいる。何